The Autism Research Institute relies on the generosity of donors like you to make this webcast possible. If you enjoy this presentation, please consider making a donation. Thank you.
Now, before we get started, I'd like to introduce our speaker. Dr. Judy Van de Water joined the faculty in the Department of Internal Medicine at the University of California, Davis in 1999. Dr. Van de Water's laboratory pursues research programs pertaining to autoimmune and clinical immune-based disorders, including the biological aspects of autism spectrum disorders. Most notable of these is the investigation of the maternal immune system as it relates to autism spectrum disorders, with particular emphasis on the presence of highly specific maternal autoantibodies to fetal brain proteins. Dr. Van de Water is currently the director of the NIEHS-funded Center for Children's Environmental Health at UC Davis, investigating potential environmental risk factors contributing to the incidence and severity of childhood autism. In addition, Dr. Van de Water's work is also part of a comprehensive and multidisciplinary analysis known as the Autism Phenome Project.
So I want to welcome everybody. I think it's great that we have the opportunity to share what we're doing here through this webinar series, and I welcome any questions. I will, as I go through this, there's some jargon, and so please, if something isn't clear, type a question and we'll try to address it at the end. Today, we work on the immunobiology of autism in both the children and the mothers, because we are trying to understand what is going on during gestation that might influence the development of the brain in terms of how immune dysregulation in the mother. And I'm not going to speak about what we're doing in the children today. Perhaps we can do that in a future webinar, but the most sort of where we are, the work that is the most mature at this point is what we're doing during gestation. So as we go through, I don't need to remind everybody of this, I'm sure that's on here, but I think important to remember now that the core autism symptoms, we're all very familiar with: the impaired social communication, social reciprocity, nonverbal and nonverbal communication, and sort of the repetitive restrictive behaviors. But what I'd really like to talk about here is thinking about all of the medical comorbidities, behavior comorbidities, genetics, and biomarkers that might be associated, that are now discussed as associated with autism. And this is from Jeremy Vandervelde at Vanderbilt, and which is, I think, is a great sort of visual on all of the different things that we know are associated with autism. But and I think now this number is down to 1 in 45, I think was the latest. And we know that it has a huge impact on society and to families. And so we're trying to address each, trying to understand the biomarkers that we might be able to diagnose earlier and come up with potential treatment pathways. So that's really what my presentation is going to be about today. And my area is, in particular, altered immune function in both the children, as I said, and the mothers, and then the mitochondrial indices I'm not going to really touch on today, although we've got some work that we're doing with Bob Navio on that. So just to give you an idea of what we do here at UC Davis, really the primary work we do is looking at environmental factors, the genes, sort of genetic susceptibility, and the immune system as they relate to autism. And we do this through population studies. We have animal models going on here, and we're all working to address how environmental triggers and immune dysregulation affect brain development, both in the animal models, and hopefully we can translate that to what's going on in people. So we look to determine how biologic markers, such as those related to immune dysfunction, might play a role in ASD, and how this may help us clarify why some children develop neurodevelopment disorders and others do not. And today I'm going to talk about the gestational immune environment in autism and how it changes in immune signaling molecules, which we are otherwise known as cytokines. You've probably heard about inflammation. Cytokines and chemokines are the molecules that we associate with inflammation, and I'll talk a little bit more about that. And how those may affect neurodevelopment. We know from animal models that these are key regulators in neurodevelopment. And then maternal autoantibodies to proteins that are highly expressed in fetal brain and their potential role in autism. So really what we work here at Davis to understand is the genes and environment and the interaction of those two. And I know there's a lot about autism genetics and trying to explain, and it's not a very large percentage of the population that we can totally attribute to genetics as a cause, but we rather look at genetics as a susceptibility. So it's that underlying underpinnings of what we see the environment reacting with, what controls how immune function is translated. You know, that's why we really look at the genetic profiling here at UCD. And the critical exposure window is very early in development. We feel that, you know, that sort of the idea that during these critical windows of neuron development, that, you know, impacting how when we impact those also translates into outcome. And a lot of this has come from work in schizophrenia as well. And I think people are starting to think that autism and schizophrenia have some parallels, especially in the maternal immune activation component of that. And then there are different clinical subgroups are likely to have different risk factor profiles. I think we're very well aware of the heterogeneity of autism and that it is not a one-size-fits-all, and we really need to see if we can define some of these subgroups through our phenotypic work. So this is our conceptual model of autism etiology. So I talked about the genetic factors as this is sort of the framework upon which everything is built. But the environment is a key factor in modifying both interrelating with genetic factors, and that includes epigenetics, and that's another area that we're working in, because these epigenetic changes also can have a profound impact on that framework. But these factors also affect immune, endocrine, and metabolic parameters, each of which we now know has some relation to the behavior that associated with autism, directly with neurodevelopment, and the medical issues, the medical comorbidities. So it's complex, but we're trying to chip away at what is going on, one piece at a time. And I'm sure everybody is very familiar with what the immune system does. It's our natural defense mechanism. It detects a wide variety of foreign agents that it responds to keep us healthy. Along with that capability of responding to this wide variety of agents comes the ability to respond to self proteins, because it's a very fine balance. And as I teach students here when I'm lecturing on clinical immunology here at Davis, it's a fine balance between having the best possible defense system and then keeping that regulated so that we don't turn that defense system on ourselves. And when that balance is upset, we end up with an autoimmune or self-reactive immune system. And so that is, I will talk about a little bit more as we get into the maternal autoantibody. And we know that nervous system, its job is to transmit signals. It's very complex. So now you've got two very complex systems that we're marrying and trying to understand what's going on in the development of autism. So it's this complex interaction between these two systems is very critical during just health and homeostasis. Normal function of the brain throughout our lifetime depends on this interaction. Response to injuries, it's critical, and part of the healing process is that inflammation that everybody talks about. And then, as I said, it's very important during early neurodevelopment. So now we've taken two very complex systems and we're marrying them to try to understand how that interaction may alter neurodevelopment. And it's such a big area now that it is actually becoming a science that we have graduate students coming in to get their degree in neuroimmunology. So we've sort of broken through some previously barriers that were previously present in sort of separating these two systems. And the key factors that we look at are cytokines and chemokines, so I'll explain a little bit more about what those are. The immune signaling pathways, and this is what's going on inside the cell. I'm not going to talk so much about that today, but those signaling pathways are really common between both systems. And so if you have a perturbation or a change in one of them, in, say, immune cells, you may that's going to translate into what's going on, and if there's a genetic change in those pathways, you're going to see it in both systems. And then antibodies, and I'm going to talk more about the autoantibodies that we have found in mothers. But first, I'll just give you a little background. Cytokines, we call them the master regulators of the immune system. The immune response is controlled by mediators, these mediators, and they're really responsible for cell-cell communication, much like what we see even in neurons. And so, you know, when two immune cells are communicating, we call that an immune synapse, which is much like the neuronal synapse. And especially when receptors and ligands are interacting. Cells produce these cytokines in response to a stimulus, so they direct the function of the cell that produces them and the nearby cells. So there's, as you can see this little picture, there's sort of a spillover effect that when anything that's in the sphere of the cell is going to be touched by those cytokines and will produce a response. They're produced by many cell types, including T cells, B cells, and macrophages in the immune system, but they're also produced by neurons, microglial cells, and many cells in the body under the right circumstances will actually produce what are typically thought of as immune cytokines. And now we're going beyond their immune function and talking about them in other developmental realms. So the immune neurointerface is just an example of how we see this interface. If we look down at the bottom, where the immune environment, these are pro-inflammatory cytokines. So the IL-1 beta, TNF-alpha, and IL-6 are really well-known inflammatory cytokines. They're what are upregulated when you're sick, especially with a viral infection. And that's their job, is to take out, you know, the invaders. But in the brain, their job really is they're very important for sleep regulation in the hypothalamus, but they're also what contributes to the fever response when you have a virus. Remember, when you get a peripheral virus, your brain knows you're sick, but it knows that because of these signaling molecules that are going from the periphery, so from the body up to the brain, to CNS. If they are in excess, we end up with a neurotoxic, sort of a neurotoxicity, but really they are important, so we need to remember that they do have a job. The anti-inflammatory pathway cytokine TGF-beta is produced to help regulate this response, so the inflammatory response, but it's also really critical for almost all aspects of neural development. So this cytokine is very important for healthy neurodevelopment, and it's produced by a lot of different kinds of cells, including immune cells, but it is a key regulator. And then we have the chemokines. The chemokines, this is just an example of some of the chemokines, but these are important for white blood cell or leukocyte trafficking, and these are they're like little chemical trails that the cells follow to get to the site that they need to do their job. So when you have, say, a skin puncture and you need to call cells in, these are the chemicals that are going to call those cells in to the site where they need to do their job. And they're also critically important for oligodendrite function, interneuron migration, and neuromodulation in the brain. So these are what control the microglial cells in the developing brain to get in there and do, say, synapse pruning, axonal pruning, sort of all the developmental processes that some of those cells do are driven by these chemical, sort of the chemical gradient of these chemokines. So immune function in autism, we know there are genes that regulate the immune response that have been implicated in autism. Abnormal immune markers in the peripheral blood of children. We've had my colleague Paul Ashwood here, and I have a lot of studies out, as do others, on changes in immune markers that we see in children with autism. Neuroglial activation and neuroinflammation in the brain and CSF. Carlos Pardo has done an amazing area of work in that area. Infection, asthma, allergies in children with autism. So there's been reports of increased and prolonged infection in some children where they just don't clear an infection, increase in asthma and allergy, which is also on the rise just in the general population. But we do see, I think one of the issues in autism is when children are affected by these, it really truly impacts their behavior. I mean, they don't feel well, and I mean, we are all irritated when we have allergies, and you can imagine if you can't really verbalize how miserable you are, it's going to have a greater impact as well. And so immune function dysfunction, are the immune changes the chicken or the egg? I mean, do they drive this change in development, or do they co-develop with changes in the brain, or are they actually a response to this? And I think we're still looking for the answer to this question, but it really is some of the basis of our research. And so one of the things we're looking at is maternal immune function. So what is going on during pregnancy or pre-pregnancy that may impact how the brain develops? And some of the things that have been looked at that I'll talk about a little bit more are increased infections during pregnancy. That's also was really first described actually in schizophrenia. Asthma, allergy, autoimmune diseases during pregnancy, sort of all of these contributing, which would be under the context of immune dysregulation in the mother, then impacting how the brain is developing. The maternal history of autoimmune disease in the family. Prior autoimmune diseases, familial autoimmune diseases have been reported. Autoantibodies in the serum plasma to fetal brain proteins postnatally and prenatally have both been described. Altered patterns of inflammatory markers in the prenatal serum and amniotic fluid, cytokines, chemokines, those have also been reported by both our group and others as potentially markers of some immune dysregulation. And I think we need to be careful when we call inflammatory markers, because those are very generic and they can be markers of a lot of things going on. Really what we use them as indicators that there's immune dysregulation, and then we can hopefully model that and figure out the mechanism by which that, how that's affecting brain development in some people. And always there's going to be an underlying, and I want to be really clear on this, there's some underlying susceptibility, because the immune system during pregnancy is geared to downregulation so that you don't reject the fetus. That's its job, is to maintain a much more regulatory state until you give birth, and then birth is very definitely an inflammatory process as you're rejecting the fetus. But to remember that changes that we see, inflammatory markers that I'm going to talk about in a minute, we're really talking about sort of changes from the norm, so what we would normally see during that time point in pregnancy and how that might affect what's going on. But as I said, this change that we have in our immune system means that many of us get sick during pregnancy because we're a little bit less protected, right? But we don't all have a child with autism because of that. So you always need to keep in mind that it's not just one thing, that there's probably multiple factors driving that outcome. So there's several unanswered questions when we talk about maternal infection and autism. Which infectious agent? Does it matter what the agent is? There's a lot of early work on viral infections, but bacterial infections might be equally at play. Parasitic infections, I think those are less in developing countries, those are less of an issue, but anything that would provoke an immune response. And then the timing, first, second, third trimester, what is the critical period? If you have the infection in the first trimester, do you have a different outcome than the second trimester? So these are all things that we can use the animal models to address. And then, is it the infectious agent, as I said, or is it the immune response to that agent? And there's been a lot of work on fever, how fever, that fever might be an indicator. If you're sick enough to actually get a fever, how long you have that fever, increased production of the cytokines directly or indirectly impacting the developing fetal brain. Is that, as I said, is it just an indicator, or are they actually having a direct effect? And then, as I said, most important, that common underlying genetic susceptibility. And I mean, I am not a geneticist, and I rely on my colleagues for that information, but we are always thinking back about what is that sort of that piece of genetic sort of susceptibility that this is overlaid on that creates a change in neurodevelopment. So these are just an example of all the papers, beginning back in 1971, Autism in Children with Congenital Rubella, Stella Chess's work, and just contributing, continuing on through 1979. So this is not a new concept. It's been out there for a while, but now we've got greater populations and a more orchestrated way of looking at these. And so now we're really, fever seems to be one of the biggest, and influences. And I remember when Useni was working on this study, he was asking me about, I was one of his co-advisors when he was a graduate student, and he said, well, I don't see a lot of influenza. And I said, well, you know, look at fever, not just influenza, because it may not be actual flu, it may be the response to that virus. And so any fever, and that turned out to be the case, that it was really more associated with fever during pregnancy than just influenza. And so we've had several studies now come out about looking at maternal infection and fever during pregnancy, but I want to remind you, and this is really important, that as I said, a lot of people get sick, and during pregnancy, and not everyone ends up having a child on the spectrum or with schizophrenia. And so there's a big difference between what we call sort of scientific significance, meaning we get a great p-value, you can see a separation in the groups, versus clinical significance. And clinical significance is when you really see that so much more, even a very high odds ratio, and way more often in the disease population than the healthy population. And so we always try to remember to keep that in mind as we are looking at reporting on our data, so that it doesn't get misconstrued. But it was an example of all the studies that have been addressing that and finding some sort of impact from infection. So I think, you know, there's a similar story that we see in maternal immune-mediated disorders or diseases, including autoimmunity and asthma and allergy. And so, you know, most studies shown some sort of association, because autoimmunity is really an indicator of immune dysregulation in a person, right? Because you don't have an autoimmune disease if you keep your immune system remains under control, and it's when you lose control over, you know, that response that you may end up with an autoimmune disease, and that would speak to either lower regulatory immune function, or just you respond so highly to something that you have a great deal of inflammation every time you respond. So increased production of cytokines directly or indirectly, how are they impacting the fetal brain? Where do autoantibodies come into this? Could we come up with an underlying, as I said, genetic susceptibility? And we're working on those questions. But some work I did with Lisa Crowan is we looked at the frequency of maternal autoimmune diseases in asthma, such as asthma and allergy, around the time of pregnancy. And what we found was that the most significant two autoimmune diseases that we saw that were diagnosed around the time of pregnancy within 2 years, either before or after pregnancy, were cirrhosis and type 1 diabetes, both of which are what we call T-cell mediated autoimmune diseases, meaning it's your T cells that really are doing the damage. Asthma was also highly correlated, as was any allergy. So in particular atopic eczema and allergic rhinitis, which is very common in the population, so that is understandable. But what this tells us is that some immune dysregulation during pregnancy, during those windows of pregnancy, could contribute to a change in immune function and resulting in changes in neurodevelopment. The timing, as I said, is critical. It was during the second trimester that we saw the highest. Odds ratio of 2.2 is not incredibly high, or 2.5, but it starts to give us sort of a picture of what might be the timing of. And the second trimester is when you're at your most immune suppressed during pregnancy, and it would be a perturbation during this trimester that may impact neurodevelopment, which is when we look at it in the animal models. And then Dr. Crowan and I have, I've worked on a study for now quite a long time now called Early Markers of Autism. This is using through Kaiser Research Group, and this is investigating early biologic markers of susceptibility and exposure from critical periods of fetal brain development. And so we're looking at during the prenatal period and then early neonatal, meaning we look at the blood spots that we get from the children here in California, the newborn blood spots. And we're looking at the etiologic, or what, the cause contribution. Is there any causal contribution from immunologic and genetic susceptibility factors, environmental exposures, and the interplay of genes with the environment in terms of autism risk. In the first phase of this study, we had a very small cohort. It was only 82 mothers of children with autism and about 160 mothers of control. Not actually, we shouldn't call them typically developing. It's really general population controls, because the only way we distinguish these two populations in this study was the ASD that sought services in the state of California, Southern California, and general population controls were women who never sought, families that never sought services. So it is not as clean a typically developing population as we have from some of our other studies, but it is a really large population. And in this early study, we saw increased levels of 3 T-cell cytokines, interferon gamma, IL-4, and IL-5, compared to the general population controls. We also have mothers of children with developmental delay without autism in this group, and they were somewhere in between. So this was sort of our first glimpse of this, and interestingly, if we look at the developmental delay population, we saw a much greater level of IL-6. And so I know IL-6 has been, through some of the animal models, IL-6 is thought to be a key cytokine in changing neurodevelopment during the maternal immune activation or infection model. But we really saw in the clinical population a much higher correlation with developmental delay without autism in that early study, in that first EMA study. So what does that mean? That increased interferon gamma, IL-4 and 5 is consistent with an allergy and asthma phenotype. This is interferon gamma is very high if you have asthma. IL-4 and 5 are contributors to allergy, sort of they drive the cells that produce an allergic response. Oops, sorry. And the placenta forms a barrier between the maternal and fetal circulation. So some get across, like IgG and IL-6, we know in the humans get across, but we are not sure about some of the other cytokines and chemokines. Even if direct passage is blocked, maternal immune components may react with the placenta and could also alter the fetal compartment. So this is something we would have to work in an animal model to better understand. So the current EMA study, which is our phase 2 study, we have a lot great. We have 400 autism women who had children with autism, 400 who were from the general population controls, and then 400 developmental delay without autism. So we had a much larger, and we could do a much better study. And what we noticed was, if you look, okay, these are the cytokines are down here on the left, cytokines and chemokines, and I've listed here what their function is, how we see their function in the immune response. If we looked, we were able in this study to segregate autism with intellectual disability from autism without intellectual disability as a subpopulation. And when we didn't really see much of a difference between all of ASD and general population controls in terms of cytokines, but what we really saw and that we thought was really even more interesting was the difference between autism with intellectual disability, meaning they had an IQ or developmental quotient below 70 or below, and autism with no intellectual disability. And you can see this long list of column of red here showing significance for a number of inflammatory, some regulatory, and then innate inflammatory chemokines, inflammatory cytokines and chemokines. We also saw a similar difference between ASD plus intellectual disability versus developmental delay without autism. So I found this really interesting because it tells me that it's very likely that the developmental delay that we see without autism has a very different development, and it has a very different profile and probably a very different gestational environment. We also saw that ASD with ID was differed from the for several of these factors from the general population controls. So it may be a nice, an interesting biomarker in terms of ASD plus ID from having a typically developing child, but really it's very nice in being able to subphenotype these two groups. So we're excited about that, and hopefully we'll be able to create a model to study that a little more in depth. So maternal autoantibody-related, or MAR, autism is another area. So I'm going to leave what's going on in gestation in terms of cytokines, chemokines, because it's an area that we're still actively investigating. That's moving forward, and then looking at sort of the better developed in our lab are autoantibodies that are present during pregnancy that recognize proteins in the developing brain. And the working model here is really, it came from this tiny little blot you can see up in the right here. We noticed that women who had children with autism had antibodies that recognized. So if I ran fetal monkey brain here in the blot, and we tested this strips of these proteins with plasma from women who had who'd had children with autism, typically developing children or children with developmental delay, we noticed that when this pattern was present, this upper and lower band, they had a child with autism. And so that we took these maternal antibodies, the production, we don't know what causes them, and that's probably the question I get asked the most, so I'm just going to get that out there right now, but we still don't know. We have, if pregnancy is even necessary. I suspect it's not from some work that we're doing now, that these may arise from other triggers, but the antibodies during gestation have a very important role in protecting the fetus. So what you've been exposed to is translated from IgG that crosses the placenta and passively protects the fetus from whatever you've been exposed to, because the fetus has no functional immune system at this point. They've had no exposures to build their repertoire of reactivity, and they are, immune naive at this point. So this is what keeps them healthy during the first 9 months of life, actually. So the placental transfer of IgG to the fetus is important, but also during this period, we know that the developing blood-brain barrier is permissive to these antibodies crossing into the brain. So we know that they can cross into the brain from our animal studies, and that if they react with proteins that are present in the brain, they could interfere with central nervous system development. So this is our working model, that we think that this causes an increased risk for having a child with autism. This is what we're now calling maternal autoantibody-related, or MAR autism. There's a lot of work behind this. As I said, we started with those early blots. We know that in terms of behavior, they're associated. The early studies showed association with regression. They are still higher in regressive, but we do see them in early onset autism, and definitely associated with language deficits, and most highly associated with stereotypic behavior. The genetics, we've looked at, working with Pat Levitt and Dan Campbell, who are both at USC now. We found that the MET variant that they described as associated in, as a higher risk of having autism in the children, we found that they are associated with the production of these antibodies. So MET is an immune regulatory molecule or receptor, and if you have this variant, you are less able to regulate your immune system. So it makes sense that it would set you up for having it easier to produce these autoantibodies. When we did MRI studies, we found that the brains of, especially in male children, prenatally exposed to these antibodies had greater total cerebral volume. And then in the basic science realm, we defined what the targets were of these autoantibodies, and I'll talk about that more in a minute. And we've got now, actually I should update this slide, we now have 3 mouse and 2 monkey models showing behavior changes after prenatal exposure to these antibodies. But interestingly, the monkey model, and I'll show this in a minute, also reported increased brain volume, especially in the males. So our model is actually mimicking what we're seeing in the humans. And then we're looking at using the tissue from these animal models to understand potential pathology in the brain. The translational potential of this is, we hope that if we can identify children with this subphenotype, we could then develop a tailored behavior treatment. We're working with behavior intervention group here at Davis now to try to stratify the kids who and what type of intervention they respond to compared to children that don't have MAR autism. Because that way we can get them, if we can test early, we can get them in there into that developmental or that behavior treatment earlier. If we could screen women at risk and develop preventative strategies, I mean, that would be a home run for us. So we have a mouse model that we're developing to do that right now. And if we can define the pathophysiology or the systems that these antibodies affect, we hopefully could come up with some maybe targeted therapeutic interventions at the small molecule level. So this is just, this whole table here is what we came out of our study where we defined what the proteins were that these antibodies recognize. And I just want to draw your attention to the top. If you react to any one of these proteins, if we have a greater percent, but it's 89% versus 70%. We live for p-values that look like this. It's a very high p-value, even a decent odds ratio. But this is what I call scientifically significant, not clinically significant. The clinical significance, meaning you could actually potentially use this to distinguish these two populations, doesn't come until we get down into these combinations of autoantigens. So antibodies that react to more than one of these proteins, and sometimes these very complex, looks like combinations, we see this is where we get more in the autism population versus the typically developing population. So this may lead us to be able to, and this is just those combinations simplified from that table, which we see in 23% of cases of women who have children with autism. It's about 23% of the population, of that population, has these autoantibodies. This is where we can start to potentially have a biomarker that could signify risk of having a child with autism. These proteins, and this is, I don't want to belabor this, but these are just all the proteins that we found that are the targets of these autoantibodies. Each has a role in different points of gestation. So neuritogenesis, dendritic branching, all the way through to axon outgrowth and growth cone collapse, meaning the maturation of a neuron. What we've seen from some of our studies, it's very neuron specific. It's not binding to other cells in the developing brain. So, and just to give you sort of a time point, the antibodies don't begin to cross the placenta until around day 100 of gestation. So they will never affect this early developmental time period. They will only affect from the time that they have access to the developing brain onward. And that the human brain consists of approximately 100 billion neurons. And I guess the word neuron would be helpful there. And during the last trimester, neurons form at a rate of about 580,000 per minute. So there are always going to be more neurons developing than we have antibodies in there. So we are not going to impact the entire brain. We're not going to impact every system all at once, but we will, we could change the trajectory of development because of these. And we've noticed that there is a correlation of some of the patterns with behavior, especially stereotypic behavior, with what we call, this is our most common pattern, which is LDH, stip and crimp. We see the biggest impact on that, and I'm not going to go into that. We also, when we did autism severity score, we noticed that boys especially have greater severity than boys that are MAR negative, so MAR positive boys versus MAR negative. Girls, on the other hand, and this is a very small number, so this is really preliminary, and we're expanding, seem to be almost more protected. Now, we haven't seen that in the animal models, but in this study, they were less severe than either of the MAR negative girls or boys. So stay tuned. This is some early data. I guess to sort of put this in perspective, we like to talk about the probability. Now, probability is not risk. It's just the probability that if you already have a child with autism, your probability of having a second child is about 17%, according to the current. So that's the recurrent statistics right now. If you have the MAR autoantibodies, your probability jumps up to 78%. And if you have a child with developmental delay, that increases to 70% if you're MAR positive over the age of 30, your probability is double from the general population, and it increases to about 35%. So this just gives you sort of an indicator of how much more likely you would be to have a child with autism if these antibodies were present in any of these groups. So what are the effect of these specific antibodies? Do they have pathologic significance? And by that I mean, do they actually physically change something during development, or are they just a really good biomarker? And so we use animal models to address this, and we have several animal models now. The first was actually a monkey model that we did passive transfer of human IgG that had these antibodies and control IgGs that did not into monkeys during gestation. Passive transfer means we purify IgG and we just inject it IV into the animals during gestation. And we saw a definite increase in stereotypic behavior and hyperactivity in monkeys exposed to these antibodies. More recently, we broke it down into those that just recognized the 3773. If you remember from that picture I showed you of the blot, these correlate with anti-STIP1, CRIMP1, and LDH. My colleague, Dr. Melissa Bauman here at UC Davis, passively transferred into rhesus macaques these antibodies throughout gestation, so 6 transfers. And the outcome is just the summary of that was mothers of the IgG-treated monkeys were overprotective. They kept their babies closer. That probably did interfere with their normal socialization. And the primatologists at the Primate Center, their take on that was that they seemed to feel that these animals felt like there was something that their babies weren't typically developing in some way, and so they were protecting them. As the infants got older, the MAR IgG treated offspring showed evidence of abnormal social behavior, both with familiar pairs and unfamiliar pairs, and they failed to engage in species-typical reciprocal social interactions, meaning they would approach another animal, but however they were behaving toward that animal, the other animal was not reciprocating their approach, and it was seen as atypical by those other animals, and we're inferring a lot by that. So we will repeat those studies. I will talk about that in a minute, but we had the ability to do MR scans throughout gestation of the monkeys and showed, similar to what we see in humans, there was an elevated total brain volume throughout this early, from 1 week to 24 months, so 2 years of age, in those animals compared to the control IgG treated animals down at the bottom. And so this is really similar to what we saw in the humans, where we know from literature and studies here at Davis that children with autism typically have larger total cerebral volume by MRI than typically developing controls. And you can see there's a lot of range here between very large and small in both groups, actually. But when we looked at those children whose mothers had the autoantibodies, it was a very tight group, and it was significantly larger brains than either population. So this is a physiologic outcome that we see in these children that actually is mimicked by what we see in the animal models. And we see the same thing in our mouse models. We see changes in social approach, increased stereotypic behavior in the terms of self-grooming, and increased marble bury, elevated anxiety. This is all with passive transfer models where we actually inject the antibodies into the mouse during gestation, the female mouse. And some interesting work with my colleague here, Veronica Martinez-Odeño, we noticed that prenatal exposure to autism-specific maternal autoantibodies changes the proliferation of the neural precursor cells, or the stem cells, enlarges the brain in these mice, and increases neuronal size, the size of the neuron in the adult brain. So at the adult brain, when it's finished developing, they have larger neurons. And so she's very good at what she does. She actually can inject. This is a pregnant mouse. They take the pups out, inject into the ventricle of the brain, which is right here. They inject the antibodies in. They put the animals back in, and they develop perfectly normally, which is amazing to me. But then we analyzed the stem cells from the fetus, and we let some grow up to be adult to do the behavior testing that I talked about a second ago. And the interesting thing for us was that if we label the antibodies before we put them in, and we can stain the brain, and this is a MAR, this animal got MAR positive, this one got typically developing IgG. You can see how much more brown there is in this one, meaning there's a lot more IgG from these, this is human IgG in these animals. So the IgG was injected here, and you can see how it's distributed in the brain. These are neural precursor cells that are developing here. This is the stem cell layer of the ventricle. So this is the part, the antibodies were injected here. They get taken up by these cells very, very nicely, actually, and you can see they're stained here. So this was really exciting to us because many of these antigens are inside the cell, many of the proteins that these antibodies recognize. And so for us, that was a really important finding because it means that these antibodies can get into the cell and find their target proteins. And the outcome of this was that it actually changes the development of these neurodegenerative cells, and you can see here this high staining. This is a control. This got the same treatment. It just got antibodies from a mother that didn't have this the MAR antigen reactivity. And you can see, I thought we'd messed up because it's so negative, and so that means the antibodies may get in, but they aren't staying in the cells. So this uptake you can see here and in here inside the cell is telling us that, and it only does this in neurons. It doesn't affect the oligodendrocytes, the microglial cells. Only in neurons do we see this. So the conclusions from this is that we think that gestational immune dysregulation may contribute to the risk of changing neurodevelopment, and this may be an underlying mechanism behind increased risk due to infections during pregnancy, so the maternal immune activation model, and I showed you the cytokine and chemokine data that speaks to that. And the maternal autoantibodies are, we think now we've done enough testing, we're really, I think, convinced that it's very specific for autism. It has the potential for clinical significance due to the very high specificity. It's only 23% of the population, but it's very highly specific when we do see it. And then the evidence that these antibodies are causing the symptoms associated with autism from our animal models is gaining strength each day. We have a new model that we've created, and I can't really talk about it till we get all the data analyzed, but this is what the different antigens, so when we identified these proteins, we then went on to identify what part of this protein and this protein and this protein the antibodies reacted to. Those are called the epitopes, and the epitope is the very tiny little part of the protein that the antibodies recognize. So we did a big array. We figured out exactly what parts of the protein that the women's antibodies recognize. We took that, we created those epitopes, those peptides, we put them into a mouse, and now we have a mouse that has created their own antibodies. We do it before we even breed the mice, and now we've created a model that makes their own antibodies that look exactly like what the humans make. And the early data are amazing. I'm very, very happy and very relieved that the data look as good as they do. We will then expand this back into the non-human primate model so that we have a more nuanced, non-human primates have so much more communication and behaviors that are similar to humans, and autism is a very higher order dysfunction disorder, right? It's communication, and the behaviors are much more, the mice, we're limited in what we can do in a mouse, so we hope that we can create the monkeys to mimic what we're seeing in the mice and expand on that. We're trying to create a biomarker test to identify women at risk, and there's a company that's licensed that from UC Davis. And then we've got prospective studies going on to determine the predictive value. So this is really important because now we're checking women prior to the birth. The original, these were associative studies, meaning that they'd already had their child, and the child had already been diagnosed. And now we're checking to see how much in the EMA study and in Marble's study here at UC Davis, what the risk of having these antibodies is. So our ongoing analysis and future directions, I've described much of the data where we are right now, and looking at the factors that potentially contribute to the behaviors. We also are looking at both prenatal and postnatal exposures to different toxicants and how that might relate to behavior. And we've got some really exciting data that's going to be coming out of here shortly on that. We're looking at genome-wide SNPs and CNVs, so variants that we might see both in the mother and the child. we sort of now, and we're combining all of these together to, you can see the complexity as it just builds here as we take each part and try to relate them together, because it's really, nothing is happening in isolation. These are connected. And then we hope in the future that we'll be able to just have a picture of at least some of what leads us to changes in neurodevelopment leading to behaviors associated with autism. So, and just to finish, why is this research important? Well, we think that if we identify patterns of maternal health conditions and biomarkers that indicate risk for a specific child of sort of changes in neurodevelopmental outcome, that will be important going forward to hopefully preventing those. And then early identification could also lead to earlier intervention and the possibility of preventing, as I said, future changes in neurodevelopment, and as well as improving quality of life. And then the identification of biomarkers for prenatal risk will hopefully shed light on the biologic mechanisms and underlying these changes in neurodevelopment, thereby providing an opportunity for developing preventative strategies. And that's always key for us, is can we intervene to change the developmental trajectory, and can we prevent so that we never have, so we don't have to worry about it. And I work with an incredible group of people, and it's a large group, and I'm very grateful to everybody and the funders who have supported all of this work. So if anybody has any questions, we don't have a lot of time left because I seem to have gone on longer than I thought I was going to, but go ahead and send questions to Denise, and I'm happy to answer any.
Okay, well, of course, we have lots of questions. So I'll try to get a few combined here so that we can hit a number of different topics. Could antibodies in a mother, strep infection, for example, be among the maternal antibodies that you're talking about? So would an infection be a maternal antibody?
Okay, so you're right. When you have an infection, you do make antibodies to that infectious agent. And for example, in pandas, it's thought that antibodies to strep are associated with changes in development in the child that has the antibodies. One of the issues that when we were looking at the epitopes, so those little parts of the protein that I was talking about that the antibodies are directed against, one of the things we're doing is we're checking those epitopes against a library of infectious agents to see if there's any similarities in those proteins because that's one of the thoughts in autoimmunity, is that infection, it's a cross-reactivity, or what we call molecular mimicry, with those proteins that directs the production that instigates the production of autoantibodies. So it's not beyond the realm of possibility that there's some kind of cross-reactivity between an antibody, say, to strep. We haven't seen that yet. It hasn't come up yet, but that doesn't mean that we won't find it. Hopefully that answers the question.
Yeah. The next question is about maternal antibody testing. They're asking about getting testing done prenatally. Is there a way to do it?
So, as I said, this has been licensed by a company that's called Pediatric Bioscience, so you can look at their website. They licensed that from the university and are developing it into a test. They're in validation right now of their platform, so they're hopeful that in the beginning of the year that test will be available. It will never be used during pregnancy, and the difference between many of the prenatal testing tests that we have now, such as Downs, is they're dependent on the fetus. The great thing about this antibody is it's made by the mother, and it can be tested at any time prior to conception. So this is really going to be, I think, we hope, useful to the high-risk community, meaning women who've already had a child with autism and desire to have a second child, that we can test them before they ever become pregnant. And I mean, it's a rule-in test, not a rule-out test, meaning you could still have a child with autism because there's many ways of getting to that disorder, but we're fairly confident that especially based on the animal models, that if you have the antibodies, that you would have a higher risk of having a child with autism. So hopefully that test will be available. I'm peripherally connected to the company and sort of keep up on what they're doing, but I don't have an exact date for their timeline. I don't know, exactly when they're planning on launching that, and it's certainly going to depend on the validation testing, and there's going to be a lot of clinical validation that they're going to do before they release it.
Okay, this next question they're asking, is there any proof of epigenetic alteration in microglia from autism patients? Have you seen any research on that?
Microglia themselves from patients we're not going to get, except in brain donors. We're looking right now at epigenetic sort of methylation patterns in cells from children with autism, especially in the context of what we call the flame retardant. We're looking at flame retardant triggers because we know they respond differently to flame retardants, and we've got some really very early data that looks promising. But I am not aware of anything looking at microglia specifically, but I know Janine LaSalle here at UC Davis is looking in brains, especially in the context of toxic exposures, for changes in methylation and epigenetic changes in brains.
Okay, this person's asking, have medical researchers using animal models like mice been able to prevent autism by changing dietary factors or using drugs?
Oh, that's a really great question, and I know there are a lot of people trying. There's not a lot of data out that's really conclusive. I know just the work from my colleague Rebecca Schmidt here, looking at folate, for example. Folate taken at the time of conception, so before conception, at conception, and early post-conception, is looks like to be a protective factor against a lot of developmental changes. I mean, we first noticed it with neural tube defects, right? That folate was really important, and it looks like it's important in autism as well. So that's one dietary factor. I think people are looking at models for stress and the amelioration of stress-induced changes in neurodevelopment of offspring through omega-3 fatty acids. I think, and I remember when I was pregnant, that was a really big deal, just in terms of they're really important for healthy neurodevelopment across the board. So a good, healthy diet in omega-3s is important. Those are the ones that I'm aware of. I think people are trying to, but they need to have the model first, right? You need to have a good model before you can fix the model.
Okay, so I've gotten a number of questions about links between mitochondrial disorders and immune dysfunction. Can you address that at all? Do you know anything about the connection between those two things?
So the job of the mitochondria is to provide energy for the cell, and the immune response is a heavy user of energy, and so when you're sick, your mitochondria have to ramp up production of energy for those immune cells that are responding, providing a response. And so that's certainly one link between the two, and if you have mitochondrial dysfunction, your immune function is not going to be as healthy, as robust, and when you do respond, you're going to really drain your mitochondria. The other issue with some of the mitochondrial factors in oxidative stress sort of manifests in oxidative stress, and those two are definitely related. Between inflammation and oxidative stress are absolutely related, and you don't really get one without the other. So that's where we're looking at those whole systems, right? Because it's important that these don't operate independent of each other.
Okay, we have time for just a couple more. I wanted to ask you this one. It's about the maternal antibody hypothesis. So if that's ongoing, they're asking, is it only a cause of inborn autism, or could it also be part of the phenomenon of regressive autism?
The very first study that we did, looking for those back before we knew what the proteins were, and we just called them the 3773 kilodalton band, we saw a very high association with regressive autism, which is a little bit counterintuitive because you would think that the early onset or sort of the inborn form of autism would be more likely in that context because it's something that's present during gestation. And I think that really, in a way that was sort of exciting to me because it means you've set up a system that whatever you're impacting may not even come into play until, say, language development or, sort of the later parts of development. And so I think it will be interesting to see as we go forward with that. The second study that we did where we determined what the antigens were and we did that population study that I showed you the big table, we saw that it was fairly high in regression. It wasn't statistically significantly higher than early onset, but definitely higher with regressive autism. So, what that means in terms of what it's setting up in terms of what other factors might influence development of autism will be really interesting to see. And some of that we can test in our animal models that we're building now.
Okay. I want to ask one more. This person's asking, how relevant would measuring systemic cytokines be in regard to reflecting possible levels in CNS? And I can't pronounce this, but I think it says parenchyma?
Oh, parenchyma. So that's a great question. Not very good. So that, I think Carlos Pardo and Susuito did a study where they were able at NIH to get CSF, and they compared what they got in the periphery to what they found in CSF. And there's not a huge correlation to what we see in the CSF, meaning the cerebral spinal fluid, and what we see in the periphery. That said, really what we have are what we can't go testing everybody for what's going on in the brain, right? So what we're having to do is infer outcome and what might be going on in the brain from what we see in the periphery, and then using our animal models to actually, and we've got an animal model right now that we are looking at if we perturb the peripheral immune system, what actually happens in the brain in terms of cytokines and chemokines, and they don't match. They definitely don't match because they're responding independently. Some cross into the brain from the periphery, some don't, and some have a greater impact than others, but we are hoping to work more of that out. But it is a great question, and it's an important question in terms of if we're going to end up trying to treat at some point on the basis of that.